Long before the word gene was even coined, curious minds began to glimpse the invisible threads that tie one generation to the next. In the mid-19th century, an Augustinian monk named Gregor Mendel tended the gardens of his monastery in Brno, in what is now the Czech Republic. Mendel carefully cross-pollinated thousands of pea plants over 8 years, tracking traits such as seed color, seed shape, flower position, and pod color. He noticed consistent patterns: yellow never mixed with green, round never mixed with wrinkled, and formulated the laws of inheritance. His 1866 paper, presenting the ideas of dominant and recessive factors, went largely unnoticed at the time. Few grasped that these units of inheritance would one day be called genes. Meanwhile, on the molecular side of the puzzle, Swiss chemist Friedrich Miescher was exploring the nature of cells. In 1869, working in Wilhelm Kuhn's laboratory in Tübingen, Miescher isolated a phosphorus-rich substance from the nuclei of white blood cells scraped from surgical bandages. He called it nuclein. He determined that nuclein contained an unusual combination of elements and was distinct from proteins, lipids, and carbohydrates. Yet, Miescher was cautious. He did not speculate on its function, and the scientific community had no ready framework to recognize nuclein's importance. For almost 50 years, genes remained conceptual and nuclein remained chemical. Most biologists assumed that the complexity of proteins, with their 20 amino acids, made them the obvious carriers of hereditary information. DNA with just 4 bases seemed too simple. But the experiments of the early 20th century began to shift this view. In 1928, British bacteriologist Frederick Griffith studied 2 strains of Streptococcus pneumoniae, one virulent, encapsulated, smooth strain, and one harmless, rough strain. He discovered that heat-killed smooth bacteria could transform live rough bacteria into deadly smooth ones. Griffith called the unknown agent the transforming principle, but he could not identify its chemical nature. A decade and a half later, at the Rockefeller Institute in New York, Oswald Avery, Colin MacLeod, and Maclyn McCarty homed in on Griffith's transforming factor. They fractionated the components of smooth strain extracts and treated them with enzymes that destroyed proteins, RNA, or DNA. Only when DNA was degraded did transformation fail. In 1944, they published their landmark paper declaring DNA the substance of heredity. The announcement generated excitement among a handful of molecular biologists but was met with skepticism by many geneticists and biochemists still wedded to the protein theory. The decisive proof arrived in 1952, courtesy of Alfred Hershey and Martha Chase. Using bacteriophages, viruses that infect bacteria, they labeled viral DNA with radioactive phosphorus, 32, and viral protein with radioactive sulfur, 35. After allowing the phages to infect E. coli, they found that only the radioactivity associated with DNA entered the bacterial cells and guided the production of new viruses. No protein radioactivity was detected inside. DNA is the genetic material of the phage, they concluded, settling once and for all that DNA carries hereditary information. With DNA's role established, scientists turned to understanding its structure. In London, Maurice Wilkins and Rosalind Franklin used X-ray diffraction to study the fine details of DNA fibers. Franklin's meticulous approach yielded photograph 51. A distinctive X-shaped pattern demonstrating a helical structure with precise dimensions. Franklin's data, shared without her direct consent, found its way to James Watson and Francis Crick at Cambridge. In February 1953, Watson and Crick unveiled their double helix model. 2 sugar phosphate backbones spiral around each other, held together by complementary base pairs, adenine with thymine, and cytosine with guanine, each pairing stabilized by hydrogen bonds. Their model explained how genetic information could be copied faithfully. Each strand serves as a template to build a new complementary strand. The publication of that model in Nature ushered in the era of molecular biology. Watson, Crick, and Wilkins shared the 1962 Nobel Prize, but Franklin's crucial contributions went largely unrecognized at the time. In subsequent decades, the race to decode genetic information accelerated on multiple fronts. During the 1960s and 70s, biochemists cracked the genetic code, the correspondence between 3-letter codons in messenger RNA and the 20 amino acids that comprise proteins. Marshall Nirenberg, Har Gobind Khorana, and others demonstrated how triplets of bases specify particular amino acids, revealing life's instruction manual in codified form. In laboratories around the world, researchers began to develop methods to read DNA sequences. In the mid-1970s, Friedrich Sanger introduced a chain termination method, later called Sanger sequencing, that allowed DNA fragments to be read one base at a time. For the first time, scientists could determine the exact order of nucleotides in genes. At the same time, the tools to manipulate DNA emerged. Herb Boyer and Stanley Cohen pioneered recombinant DNA technology in 1972, using restriction enzymes, molecular scissors that cut DNA at specific sequences, to splice genes into plasmid vectors. Bacteria transformed with these plasmids could express foreign proteins. In 1978, Genentech used recombinant DNA to produce human insulin in E. coli, setting the stage for modern biotechnology and ushering in a new era of pharmaceuticals. The 1980s saw automation transform DNA sequencing. Multi-channel fluorescent detectors replaced radioactive labels, and sequencing machines accelerated the process. These advances, coupled with the invention of the polymerase chain reaction (PCR) by Kary Mullis in 1983, revolutionized molecular biology. PCR made it possible to amplify tiny amounts of DNA millionsfold in a matter of hours, enabling forensic scientists to identify individuals from minute biological samples, clinicians to detect pathogens rapidly, and geneticists to study genes in unprecedented detail. Fueled by these technologies and political will, the Human Genome Project, HGP, was launched in 1990. A public-private consortium of universities and government agencies across 6 countries set out to sequence the entire 3 billion base pair human genome. Using shotgun sequencing and hierarchical mapping approaches, they tackled chromosomes one by one. In parallel, a private effort by Celera Genomics, led by J. Craig Venter, employed faster whole-genome shotgun techniques. In June 2000, a joint announcement from President Bill Clinton and Prime Minister Tony Blair unveiled draft sequences from both projects. By April 2003, 2 years ahead of schedule, the high-quality reference human genome was declared complete. The HGP revealed that humans have roughly 20,000 to 25,000 protein-coding genes, a far smaller number than previously imagined, and vast amounts of non-coding DNA whose regulatory and structural roles are still being unraveled. Armed with genome sequences, researchers launched functional genomics initiatives like ENCODE, Encyclopedia of DNA Elements, to map gene regulatory elements, non-coding RNAs, and chromatin states across cell types. They discovered microRNAs, small regulatory RNAs that fine-tune gene expression, and long non-coding RNAs that scaffold chromosomal organization. Epigenetics, heritable modifications such as DNA methylation and histone acetylation, emerged as crucial layers of gene regulation, explaining how identical genomes could produce diverse cell types and how environmental factors might leave marks on the genome that influence health and disease. The 21st century brought another revolution: next-generation sequencing, NGS. Platforms from companies like Illumina and Ion Torrent leveraged massively parallel sequencing, reducing costs by orders of magnitude and enabling whole genome, whole exome, and transcriptome sequencing on a personal computer's worth of data. NGS fueled large-scale projects such as the 1,000 Genomes Project, which cataloged human genetic variation worldwide, and the Cancer Genome Atlas, which profiled mutations across thousands of tumors to guide precision oncology. In 2012, a breakthrough in genome editing arrived with CRISPR-Cas9. Drawn from a bacterial adaptive immune system, CRISPR clustered regularly interspaced short palindromic repeats, and its associated Cas9 nuclease were repurposed by Jennifer Doudna, Emmanuel Charpentier, and others to cut DNA at programmable sites defined by a short guide RNA. Suddenly, researchers could edit genes in living cells with unprecedented ease, precision, and efficiency, knocking out genes, correcting mutations, and inserting new sequences. CRISPR opened doors to potential therapies for genetic diseases, agricultural improvements through gene-edited crops, and the resurrection of extinct traits in endangered species. Ethical debates swirled as the possibility of human germline editing, designer babies, and ecological disruption came into view. Simultaneously, single-cell genomics emerged. Researchers combined microfluidics and NGS to sequence the transcriptomes and genomes of individual cells, revealing cellular heterogeneity in development, immunity, and cancer. Spatial transcriptomics added another dimension, mapping gene expression within intact tissues. Meanwhile, nanopore sequencing using protein pores to read DNA strands directly promised real-time, portable sequencing in the field, from remote disease outbreaks to space missions. Today, DNA science underpins fields as diverse as synthetic biology, where scientists design and assemble entire genetic circuits or even minimal bacterial genomes, to paleogenomics, which recovers and sequences DNA from Neanderthal bones and mammoth tusks, rewriting our understanding of human evolution and ancient ecosystems. The Human Cell Atlas aims to catalog every cell type in the human body, while global initiatives like Earth Biogenome plan to sequence the genomes of all eukaryotic life on Earth. The story of DNA is far from complete. As researchers explore three-dimensional genome folding, phase-separated nuclear compartments, and the interplay between genetics, epigenetics, and the environment, they confront new mysteries. How do non-coding regions orchestrate complex developmental programs? What unforeseen consequences arise from genome editing? How can we ensure equitable access to genomic medicine? And perhaps most profoundly, how does the intricate dance of four simple nucleotides give rise to the rich tapestry of life on Earth? From Mendel's Ps to CRISPR's precise cuts, from Miescher's muddy bandages to the high-throughput sequencers of today, from the first glimmers of nuclein to the ongoing quest to rewrite life's code, DNA's story is one of human curiosity, ingenuity, and collaboration. It is the story of our pursuit to understand who we are, where we came from, and what we might become. And as we continue to probe the depths of the genome, we write new chapters in the epic saga of the molecule that binds us all. February 2nd.